Hello students, so this is our next class. At previous class we have studied about signaling. Here NRTK or different pathway of NRTK. In today's class we'll study about the uh, bacterial cell signaling. Okay, so what is two component system? Its primary definition is the primary signal pathway for sensing environmental changes, and it is what its importance. It is crucial for survival, adaptation, and virulence regulation. So here is the two comparative diagram where in resting phase and activated phase are been shown. In resting phase, a sensory histidine kinase is present. That is a transmembrane protein that detects external stimuli. And what is response regulator? It's a cytoplasmic protein that uh, mediate or transport cellular response to the gene expression, DNA, etc. So in resting phase, these both are present, and in activate state, when stimuli bind to the histidine kinase receptor, autophosphorylation or self-phosphorylation will occur. Through this phosphorylation, it activates the response regulator, and response regulator will express in DNA. Then how it's act? How the regulation of two component system occur? First, autophosphorylation. When the stimuli bind to the or ligand bind to the receptor, autophosphorylation of the uh, cytoplasmic end in the cytoplasmic end, autophosphorylation occur. After that, it stimulates the RR, the response receptor, response regulator. After the response regulator activate, it's travel to the nucleus and bind to the DNA, express or then express gene or alter the gene expression. After that, the phosphorylation remove by phosphatase, so that it uh, again again come into inactivated state. As you know, before we have studied the desensitization pathway, where continuous activation of some regulator response occur, then desensitization will occur to pause the expression. Then regulation of osmolarity. Here, the sensory kinase receptor ENVJ, after activation, it promotes the OMPR in the DNA, and this OMPR gene expression promote or stimulate or activate OMPF and OMPC. How this channel activate and why this channel activate? When low osmolarity is present in the environment, low kinase or low OMPRP will occur, and OMPF will open. During high osmolarity or salty nature, high kinase or high OMPRP activate and stimulate the OMPC, which restrict the entry. What is bacterial chemotaxis? Taxis. What is, its definition is direct movement in response to chemical stimuli. What is receptor 2? It is a methyl accepting chemotaxis protein which cluster all the cell pores to detect gradient of the signal. But then the receptor bind initiate intracellular protein related to the flagella motor. After signal binding, the receptor activates and it stimulates the flagella to move clockwise and anti-clockwise. Then the expression through that the cell will go toward the response or away from the response. Here, move toward the attractant if the signal means nutrition, or away from the replicant if it is toxin. What is the run and tremble mechanism of the bacteria? Did you know it? The flagella move counterclockwise. Flagella bundle for straight swimming in presence of uh, nutrients or etc. If uh, when it's tremble during flagella rotate in clockwise direction, flagella fly or apart or random reorientation so that it does not moves toward the receptor or signal of, or it has a toxin. Structure of the bacterial flagella. Here motor A and motor B, a form of stato or to power the rotor. What is gram negative? Sorry, in gram negative contain 4 rings spanning outer membrane, peptidoglycan and inner membrane. In gram positive contain only 2 rings due to lack of outer membrane. During the activation or signal, if it is nutrient, then it activate and move anti-clockwise so that uh, it move toward the signal, and in presence of toxin it's move clockwise so that it trembles and go away from the toxin. They all are being activated by motor A and motor V so that there is flagella move or rotate. What is the chemotaxis protein and their functions? When the receptor activate and it's autophosphorate and activate the CHEA, CHEA moves in the cytoplasm and activates CHEY. Through it activate the receptor motor A and motor B, mod A and mod B, where it move clockwise rotation or anti-clockwise rotation. Here in absence of signal and here in presence of signal. In presence of signal, CHEA doesn't activate. That's why CHEY activate and bind to the rotor or flagella, move towards the replicant, nutrients. Mutation in the chemotaxis protein. When CHEY lost the function, cannot relay signal to the motor. During this, smooth swimming continuously occur or move toward the signals it occur continuously. CHEA lost the function, then cannot phosphorylate CHEY. Due to in absence of CHEY, again the smooth swimming occur. When CHP lost the adaptation, cannot demethylate MCP stock off and the receptor remain off. Due to their trembling phenotypes occur. CHER lost the methylation, cannot methylate MCP stock on. So smooth swimming occur and move towards the signal. Mutation in specific CHE protein result in predictable observable swimming phenotypes. So as for today, we'll study about the signaling, and day after tomorrow or tomorrow we'll study about quorum sensing. So thank you, and read all this, try to clear gate, CSIR, etc. Okay, bye.